Hi, I'm Ashley Volz. I'm here on site at the RNA Leaders event in Basel with the director of Asymchem. Thanks for meeting with me today.
Hi, nice to meet you. Myself, Arumugam Balasubramanian. I am working as a director at Asymchem. Asymchem is one of the top CDMO companies in China. I'm very happy to be here for the interview. Thank you.
Great. Yeah, it's great to meet you. Can you tell us the control strategies needed to focus on the manufacturing of oligonucleotides?
The raw material quality, water content of the solvents and reagents which we prepare for the coupling reaction, and the amount of the solution which we prepare for the coupling reactions needed to be surplus amount by considering the dead volumes of the container and the dead volumes of the pipelines for the solid phase synthesis. Then the DCA concentration needs to be considered in such a way to avoid the depurination impurities, and after that on the cleavage and deprotection side we need to do the workup process as quickly as possible in order to avoid the decomposition of the product during the longer contact time of the solution. Then on the purification side we need to consider the loading amount of the material, after that the fooling strategy of the purified fractions, and after that the lyophilization parameters need to be developed before we go for the large scale lyophilization. So these are the control strategies we need to take care during the manufacture of oligonucleotides to get the oligonucleotide with high purity and high yield.
Interesting. Okay. And what technologies has Asymchem developed for the improvement of oligonucleotide service?
For the most of the oligonucleotides are synthesized using solid phase synthesis, in that there are large amount of phosphoramides and solvent usage. In order to avoid that, we have developed 3 more technologies like liquid phase peptide synthesis, liquid phase oligonucleotide synthesis. There we can reduce the amount of solvents as well as the use of phosphoramides equivalents, and after that enzymatic ligation methods. In that enzymatic ligation we prepare the fragments, and then after that we do the annealing and the ligation by in liquid phase. For doing the ligation we have more than 300 ligases in our library that can be used for the client project without having any issue, and the material we have got with the technology we have developed through the enzymatic ligation technology. We can be able to get the material with the high purity like more than 95% by controlling the N- and N+ impurities. After that we have also developed the Blackmer technology. In that dimer and trimer will be prepared, then they will be coupled on the solid support. Finally we will do the cleavage and deprotection purification followed by lyophilization. By this technology we can be able to shorten the coupling time as we are preparing the Blackmers, and also we can be able to control the N- impurities, because N- impurities are closer to the main peak. They are very difficult to remove during the purification. So in this way we can be able to reduce the impurities and improve the yield.
Great. And what makes Asymchem different or advantageous over other CDMO companies?
We have experts with experience between 5 to 10 years on oligonucleotides synthesis and development on both the process as well as in the purification side. Then we have manufacture equipments from small scale to large scale. With that we can be able to make the oligonucleotides up to 1000 kilogram per annum. Then we have developed different technologies other than solid phase synthesis. In addition to that we also have the process for the preparation of phosphoramides. Phosphoramides like natural ones, chemically modified ones, and also custom made ones. These amides are prepared with the quality comparable with the quality which we are obtaining from the routine suppliers. In that we can be able to provide the material with a competitive cost in a shorter lead time. So through this way we can provide the service to the customers to reduce the cost and timeline. And then we also have the equipment, the analytical instrument facility. With that, we can be able to do the full release testing and stability testing without going for the third party analysis. And finally, we have the solvent recovery technology. When we go for the commercial scale manufacturing, the solvent which we are using in surplus amount in the solid phase synthesis can be recovered with the technology we have developed from the small molecules. And finally, we also have the late phase experience from the small molecules that can be used for the oligonucleotide late phase programs. In addition to that, we also have the experience on the oligonucleotides NDA projects, about 4 numbers.
Great. Well, thank you so much for meeting with me today.
Thank you.